So today our first speaker is Natasha Caplen. She got her PhD from the University of London, King's College. And in 1996, she came to the National Human Genome Research Institute at NIH. And CCR, we thought she was really smart, so then we hired her in 2004 as a senior scientist. She heads the gene silencing section, and she is now in the genetics branch. Her title, Functional Genomics via RNAi. Natasha.
Thank you. Okay, everyone can hear me in the few people that have been able to brave the storm and weather on a wet November and Monday. But welcome to those here, and I gather there are lots of people online or will be watching this in the future. So as Terry said, my topic today is functional genomics. I'm going to be concentrating on what I think of that in the context of RNA interference. But at the very end, I'm going to touch on how other technologies are being developed that complement RNA interference-based work. So today's presentation, I'm going to give you a brief introduction to RNAi and RNA-based technologies. There's not really a quorum in the room where I can make sure this survey is valid, but I'm going to do it anyway. I just want to get a sense from the few people that are here in the room, how many of you have done an RNAi-based experiment in the lab? Got 3 plus Dr. Moody. Okay, so about 50%. Whether that translates out into the wider world, I'm not sure, but that gives me some sense of where we're at. So I'm going to give an introduction to RNAi and RNA-based technologies. Then I'm going to give you a few real-world cases, and these are cases where these are projects that we've conducted in collaboration with various investigators within CCR that have been focused in my lab, that I've chosen to try and illustrate some of the key features of how we apply RNAi to study gene function. So I'm going to talk about breast cancer cell survival. I'm going to talk about genes that are amplified in colorectal cancer. I'm going to talk about a study that's also in breast cancer where we looked at TRAIL-induced apoptosis. And then I'm going to move on to just a couple of brief slides talking about genome-wide RNAi screening. I understand from Dr. Moody, you saying you've been to NCATS or you're going to NCATS? Yeah. You've had a tour, so that will bring into NCATS at that point on the work that they're doing in the RNAi screening area. And then, as I say, very briefly at the end, I'm going to talk about other functional genomic approaches. All right, so I think RNAi is a great fit for this particular set of presentations and this particular course, because this really has been truly a translational research story. This is something that started in plants and fungi and now is in clinical trials and is being used for drug discovery targets, for analyzing drugs, really in a very translational setting. So, as I said, it was originally described, what we now know as the phenomena of RNAi was really described in the early 90s in petunias and in fungi. So it was seen as a post-transcriptional gene silencing mechanism. So it was observed that when you manipulated plants or fungi in a particular way, you saw suppression of gene expression. But it wasn't until the late 1990s and the work of Andy Fryer and Craig Mello that won them the 2006 Nobel Prize that we actually understood that what was the mediator of this phenomena was double-stranded RNA. And you saw in work that I did, and then also Tom Torshall's lab that was then at the Max Planck, looked at how we could actually induce this phenomena in mammalian cells. And then this is really where I came into this field. People have then taken on this into clinical trials, or say into using it as a very robust and well, broadly widely used tool for studying gene function. Okay, so I'm not going to go through the mechanism of the RNAi pathway in a great deal of details. I'm just going to give you a few highlights so that everybody's on the same page. So RNAi is not there just as a really fun tool for us. It's there as a critical pathway for regulating gene expression. And the best mediators of RNAi in mammalian cells, or the best characterization, are mature microRNAs. So this entire pathway uses small RNA molecules of about at the end point of about 20 to 27 nucleotides. So there are some that are larger, but I'm going to focus on the species that are around 21, 22 nucleotides. So in microRNAs, you have an entire pathway that is developed that involved in the biogenesis of microRNAs. These are expressed from genes that are within, look like standard genes, but they're non-coding. So these are regulatory RNAs. And these are involved in the post-transcriptional gene regulation of between 30 and 50% of protein encoding genes. And so this in itself is a major topic. I think there's at least one person that talks about some of that within this course. So I'm not going to talk about this beyond today, other than saying that this is the mechanism that we're exploiting when we conduct RNAi-based technology experiments. So over on this side, we have gene regulation, and over on this side, we have loss of function analysis. Now, what is the key difference between these? Well, in the end, the key difference between these two is actually related to the sequences of the molecules that you're using. So microRNAs traditionally have a interact with targets, mRNAs, in the 3' UTR and have mismatches in the alignment of the sequences of the mature microRNA with the sequence that it targets in the mRNA. So there is an inexact match. Whereas siRNAs that we're designing for functional genomic analysis, they have to have a perfect match sequence. And this actually drives two different processes. Here, what you're seeing is predominantly an effect that reduces mRNA stability, leads to mRNA degradation and or translational repression. This is going through one group of the Argonaut proteins, which I'm not going to go into detail about, but this is where you have, as I say, generally a transcriptional repression as a consequence of mRNA degradation and changes in mRNA stability. Through the siRNA pathway, however, we are exploiting a feature of just one of the Argonaut proteins, Argonaut 2, and an exact match between the siRNA sequence and the target transcript. This induces a transcript cleavage at a predictable site in the sequence. This then is degraded, and you see your subsequent loss of protein production and loss of function. I'm going to come back to this in a few moments when I discuss how we have to understand the differences between these processes if we're truly going to interpret data, because siRNAs can act as microRNAs if you have a sufficient sequence alignment. And this is often referred to as off-target effects. And I'm going to mention that briefly, that you have to take that into account when designing any experiments using RNAi-based technologies. So why do we, why has RNAi become so important? I think one of the reasons is that, particularly in the context of cancer cells, our ability to profile the cancer genome on a very large scale has really been very much revolutionized over the last few years. We know we can do large-scale epidemiological studies to get gene association. We can get genome copy number and epigenetic analysis on a genome-wide basis. We can do sequence analysis and gene expression analysis on a genome-wide basis. But systematically functionalizing these findings, finding out what this actually means, is still a significant challenge and is very much a bottleneck in really making maximum use of all the information we're getting. And so one of my remits over the last few years has been trying to think about how we can both do genome-wide, as I'll mention later, do functional analysis on a genome-wide level, but also coming down to individual genes to make use of that information. And I'm going to talk about both of those today. So again, just as a brief couple of introductory slides. So when I talk about RNAi analysis, we're talking about what is still probably the most widely used approach for conducting gene-specific and loss of function studies. And so we are inducing a loss of function. So we're comparing two populations where we have eliminated a gene, which is shown here. So what we've done is we have the siRNA that's targeting the target transcript with exact sequence match. This is inducing the target cleavage by AGO2. We inhibit protein production. We have a loss of some sort of cellular function, some sort of phenotype that we can measure. We are inhibiting protein production, and if a protein is very stable, then you may not induce a phenotype for some time. And so there are different ways of inducing RNAi, either by using an siRNA, which is a transient effect, and what's predominantly what my laboratory has used, which means you need to see a phenotype, you have to induce something over about a maximum of 5 to 6 days. Or you can use an shRNA, which is basically has a similar physical characteristics of a microRNA in that it's a hairpin, and these can be expressed within a plasmid and can be actually stably expressed, at least in some circumstances. So what are people using RNA-based technologies for? As I say, you can use it for standard gene function analysis, but more importantly, it's been moving up to using it for pathway and network function, molecular target identification, which is what I will talk about today. Talk about how a compound is working, or trying to find ways of combining two drugs together to improve the efficacy of both of them. And then also we've used it in the context of biomarker validation, so I won't talk about that today. Okay, so in the lab, when we're developing an RNAi-based experiment, there's a number of things we need to consider. Probably the most key is the model system that you're going to ask the question in. So as you're going to see going forward, what we've done is a number of studies where we have conducted RNA experiments, we've silenced a gene, but the only way we could truly interpret what the function of a particular protein was by remembering what the context is of the question we were asking. So if we're studying in breast cancer, we need to be careful that we're looking at a full range of different breast cancer cell lines. If we're looking at colorectal cancer, we need to look at this in the context of a range of colorectal cancers, because that's the only way to really be able to truly interpret the data that may be coming from tumors that are, by definition, often heterogeneous in their genetic background. Okay. So absolutely key to everything that we've ever done, to be honest, has been making sure we choose the right model systems to do this. And then we have to optimize how we're inducing RNAi so we ensure we've got high efficacy and high specificity. So as I mentioned earlier, we can use siRNAs, which is what my lab traditionally does, but we can also use shRNAs to induce RNAi. Then we need very good assays, and I cannot stress enough that this step, and over the years I've worked with a great many postdocs particularly who've saying, my siRNA doesn't work, and the answer is no, usually it's your antibody that doesn't work, or your QRT-PCR primers. This step is just as important as in defining the model system. And then obviously having a phenotypic readout is critical. So I'm just throwing in a couple of really now quite old slides, but just to give you a sense of what a successful RNAi experiment looks like in my lab. So this is actually looking at mRNA expression for a very well-known gene involved in colorectal cancer, but in others, beta-catenin is part of the beta-catenin Wnt signaling. And what we're seeing here is a measure of RNA levels, which is being normalized. And then we have here an Si negative. So this is a control siRNA. There are a number that are commercially available. These siRNAs have been defined so that they have sequences that do not match to anything within the mammalian genome, any transcriptome within the mammalian genome, at least for protein encoding genes. These are not scrambled, and I'm going to come on to this. These are actually very well documented controls, and I can strongly urge that you look at those. We would often also run, in something like this, an untransfected control. So we're making sure that the untransfected and the Si negatives look exactly the same. And you're going to see some of that in some of the more complex experiments later on. And then we do a minimum of at least 2 siRNAs corresponding to every gene that we look at. As we move down through a complicated study, we might eventually end up with just looking at 1 siRNA. But the study at the moment, for example, that we're writing up at the moment, we started with 6 siRNAs against multiple genes. And as we go through the paper, we go down to and we get to more complex phenotypes that we're measuring. We might get down to 1 or 2 siRNAs, but we'll always start with a large number. And that's so that we're very robust about what we're interpreting and what we're seeing. So if a paper from you with 1 siRNA goes across my desk, it gets immediately rejected. So I would not advise me as a reviewer under those circumstances. So this is then looking at a protein level. And there's an important take-home message from this is that what we've done here is we've looked at the knockdown again of beta-catenin with 2 different siRNAs, but we've done it over time. And this comes back to that issue that what we're looking at is the blockage of production of protein. So this will be dependent, where you'll see a phenotype is going to be dependent on whether the protein stability is such that it becomes degraded enough that you now no longer see that protein. And so we often have to do, and I make people do very complicated time courses so we know exactly at which point we should be looking at start to see a phenotypic effect. Some proteins degrade very, very quickly, and you'll see an effect within 24 hours. Some do not. Some you may have to go out for several days before you'll see an effect. So this is very important to consider when looking at any particular protein of interest. And it actually astonishes me still, one of the critiques we've had when we've often talked with, again, usually postdocs at the NIH that come talk to us about it, I want to do an experiment, and say, well, what's the half-life of the protein you're trying to silence? And that is just information that they either don't know or is just not available. And we'll have to say you're going to have to figure that out because you've got to know when to look. It's very important. Okay, so I'm now going to touch back to a point that I made a little earlier, and these are just all the different features that you need to consider when you're thinking about how to design RNAi experiments. And this comes back to this question of off-target effects. So I said earlier that actually an siRNA can act as a microRNA, and that's because the microRNA is a key portion that interacts to find a particular target within the 3' UTR of a protein encoding gene is actually just the first 7 to 8 nucleotides on this end of the microRNA. And this is known as the seed sequence. And so if an siRNA happens to have a seed sequence matched to another protein encoding gene in the 3' UTR, it can have an effect on those targets. So that's why we often use multiple siRNAs. They will have different targets, and if you see a consistency of phenotype, you're going to have more confidence that you're seeing an effect. But I will mention other ways that we can look at this as well in a moment. One of the key things is also, so some of the features of how to design experiments are sequence independent and some are sequence dependent. I've just described here something that is sequence dependent, so the silencing of an unintended target through partial complementarity. So you want to use, to control for this, your RNAi effector, your siRNA or shRNA at the lowest possible dose. You want to use negative controls, and you need to use multiple effectors. And that's the easiest way to address this. There are some other features that have been described in the literature, but you just need to be aware of. You can saturate the endogenous RNAi machinery, and in some cell types and in some circumstances, you can induce an immune response. And this is relatively well described in the context of people that are trying to develop RNAi therapeutics, but for most people, it's not going to be an issue in the lab. Now, I'm just going to briefly talk about one way that actually a former trainee in my lab who actually runs the trans-NIH RNAi screening facility at NCATS has tried to think about, in a very clever way of trying to think about how we can control further for these siRNA microRNA-like interactions. And this is a busy slide because I pulled it from their really quite complicated paper, and I really just wanted to have this so you had it as a handout for future reference, is that what we're looking at here is this key figure here. What they've learned is that if we mutate just 3 nucleotides in the center part of the siRNA, known as the positions 9, 10, and 11, what you can effectively do is make a sequence-specific control for each of the siRNAs that you're studying. And you can then use that to determine whether you've got a false negative or a false positive. And so a lot of people in the field are starting to adapt to using this, particularly for facilities where they want to confirm a large number of hits from a particular screen. And so I just put this for you to consider if you're wanting to have additional controls for any additional experiments. So what I'm now going to do is turn to some of the cases where we've actually applied RNAi in the lab using thinking about some of the features that I've just talked about, making sure we have really good assays in place, that we're using lots of different RNAi, and that we're using the right models. So what my lab has done over the last few years is sort of summarized in this one slide. What we've done is we've developed siRNA in a way that we can screen these with an individual siRNA in each well of a 384 well plate with all the appropriate controls. And then we can do lots of different assays that are very, very similar to what you would do in the context of drug screening. So in effect, what we've done is instead of now using drug inhibitors, we're now just knocking out every gene up to the genome, as I'll describe at the end. So assays that we've run routinely in my lab are particularly cell viability. It's very, the cancer community is very keen on killing cells. That's very much a key parameter for them. But we've also done, as I'll talk about, studies in apoptosis and also reporter activities. Then what we've done is then, if we've had an individual or few genes that were of interest, we've then gone on to do very comprehensive molecular and functional analysis of that particular hit. And in some cases, we've also combined either RNAi screening and our RNA analysis approaches with a particular therapeutic intervention. So where we've been trying to study conduct drug analysis in combination with RNAi. So in the first case I'm going to talk about is in breast cancer, and the aim for this was the identification of proteins required for cancer cell survival. And our hypothesis being that if we could do this, we may be able to find new cancer therapeutic strategies. And so this is obviously a broad aim for a number of labs within CCR, and it's a sort of a broad aim for a lot of screens that have been done in the RNAi field. So just to make sure everyone knows what we're talking about, so breast cancer, we know it's a very heterogeneous definition for a group of cancers, in effect, because now we know that the molecular basis for different breast cancers, depending on the site of origin and the genetic makeup, can be differentiated. So some of the three most general definitions that you can have are tumors that have are ER and or PR positive. Then you have amplified ERB2, and then you have what are often known as the triple-negative breast cancers, which have none of these changes. And these are important because this very much defines now how breast cancer is being treated, whether you're going to be able to attack the dependence of these tumors on estrogen, whether you can use anti-ERB2 treatments, and then if you're triple-negative, really the options are really only surgery and chemotherapeutic. So there are no targeted treatments here, and this is one of the things that we've been trying to work with a number of collaborators over the last few years to try and attack this problem. So there's critical need for molecular target therapies in all of these. None of these are perfect. We always want to know, but there was a particular emphasis in this project, at least at the begin with, in triple-negative breast cancer. So this was a study that was started. It was a collaboration with Stan Lipovitz, who's now a branch chief for the Women's Malignancy branch in the Center for Cancer Research. And it was started by an incredibly talented pre-ERTA. She went to grad school, and I'm sure has done very, very well. I've lost track. And a postdoc in Stan's lab. And what we're seeing here is where we have silenced in triplicate, we've done RNAi screens against the human tyrosine kinome. This was the human tyrosine kinome, kinases were a major focus of Stan's work, and so we really focused down on that. And this was done in the triple-negative breast cancer MDM231. And so what we're seeing here is we've scored the relative viability of this breast cancer cell line versus a known control. And this was a gene, was siRNAs against the gene RM2. RM2 is a component of the ribonucleotide reductase. Every cell is completely dependent on the expression of the ribonucleotide reductase, because without it you can't make DNTPs and you can't make DNA. So these will die. So this was sort of our maximum effect. So if we could find any proteins that when we silenced them gave an even better effect than RM2, we knew we were onto a potential winner. And so what you have here is his RM2, so we've scored everything as when these genes were silenced, we see an effect on the growth of these cells, and here actually these cells are either inducing are not as, they are reducing viability, but not as much as sort of our positive control in this context. So the gene that we ended up following up on is at the bottom of this list, and that's WEE1. Here we're looking at it in more detail, and now we've moved from one cell line into 3 breast cancer cell lines, and then a non-tumorigenic mammary epithelial cell line. And this was very key to the success of this study, and is not always feasible in the context of cancer research, as I'll mention in a few minutes, but was very important for this study in that we could compare this to a line that grows in culture, we can transfect, but is non-tumorigenic. It has a P16 mutation, but it doesn't, so it can grow, but it is basically non-tumorigenic. So if we can find something that has an effect on the growth of these cells, but not these, we may have a differential effect, and that's a selective effect, so that's important. So what I hope you can see here is we have silencing. We see when we silence, and this is just showing data for one siRNA, we see a reduction in the growth of these cells, but not in these cells. And here we're seeing the silencing of WEE1 at a protein level in all cases. So we know we're silencing WEE1, but there's no effect in the MCF10As. So what is WEE1? Well, it's absolutely critical in the terms of the regulation of the cell cycle. So entry into mitosis is regulated by the cyclin-dependent kinase complex, and this includes a serine-threonine protein kinase known as CDC2, to show here. And CDK activity is then held in check by the inhibitory phosphorylation of CCD2 by WEE1. Okay? So if you remove WEE1, you are probably altering that. So entry into mitosis is dependent on the protein phosphatase CDC25B. It removes this phosphorylation from CDC2. So there's this relationship between these. WEE1 puts it on, CDC25 puts it on, and that activates to go from G2 to M. So we can actually look at this at this level, and what was key was that fortunately an inhibitor of WEE1 has also been generated, and we were able to, with the drug, to now move to even more cell lines, and we're able to show that actually all of these breast cancer lines is representing ER positive, HER2 amplified, both forms of triple negative, whether basal A or basal B, which is based on gene expression profiling. All are sensitive to the inhibition of WEE1 with a particular drug. I'm not going to go through its whole name. But MCF10As are not. And what we see is that it inhibits the phosphorylation of CDC2 in all cell lines, but only reduces the viability of the breast cancer cell lines. So now we've been able to take an RNAi experiment and now confirm that pharmacologically. And what Lindsay and Sarisha were able to prove was that this is actually, if you look at DNA damage, the WEE1 silence or inhibition, they show induced DNA damage, but only in the cancer cells. So we now have a basis for the mechanism of this, which related to how WEE1 is required for the cell cycle. And actually, another functional genomic screen doing actually an siRNA screen found a similar effect in another triple breast cancer cell line in the same year. So what we found was we identified WEE1 functions required for the viability of multiple cell lines, but only breast cancer cell lines and not a mammary epithelial non-tumorigenic line. And so this is potentially consistent with the concept of synthetic lethality, and that suggests that the breast cancer cell lines used in the study are selectively sensitive to WEE1 loss of function. And so a number of groups are now actually following up on this and other observations that a number of groups have made about WEE1. So for this purposes, for this presentation, what I wanted to get across to you is how we were able to take a sort of, we focused down on one group of very important proteins, kinases, and then we're able to go down to one protein that we think has a critical but selective effect. But to do that, we had to be able to take it from one cell line to a number of different cell lines. And very importantly, we're able to also use a drug to confirm those data. And we've used that consistently through our work, that where possible, if there's a drug that we can confirm with, we will. So I'm now going to go into a slightly different case study, but where we've, instead of doing we've done a small, even more focused screen, but have really focused on the comprehensive molecular analysis downstream of a limited number of genes to really understand why a particular set of cells might be dependent on on that protein for cancer cell survival, and use it to really understand potentially both the normal and cancer-related functions of a protein. And this has been done in the context of colorectal cancer. So this is, as you know, is still a major cancer in terms of morbidity and mortality. So there are about 150,000 colorectal cancer diagnoses in the US annually, and even with improved screening, we need to have a better understanding of this disease to develop new therapeutic strategies. So a lot of the molecular changes have been well characterized, particularly in the early stages when you're going from so from polyps to a full cancer. But in advanced cancers, colorectal cancers, there's still a lot of room to understand what are some of the changes that are occurring at this point. And this is what my collaborator, Thomas Reed, who's actually in this building over there, from the genetics branch, has been studying for many years, and I've done a number of studies with him over the last few years. And this was from a paper that we published last year in Cancer Research, led by a postdoc in Thomas's lab, Jordi Camps, and then by a CERTA fellow in my group, Jason Pitt. And so what they came to me, and we had to go through, I've put down here, these were all the steps that we had to do to get this paper to work, get this study to work. So what they came to me with was originally this concept that there were a lot of candidate genes in a particular region that showed copy number changes and changes in gene expression that they wanted to study. And I'm going to go through that in a little bit more detail in a moment. We then needed to validate what was our model that we were going to study these genes in. We then screened these. We did 2 screens. Then we did the molecular profiling, and then we did a hypothesis generation and follow-up on just one of these genes. So the concept that they came to me was that in advanced colorectal cancer, you almost, or in fact, Thomas would say, I'm not even allowed to make that vague statement. You always see amplification of regions of chromosome 13 in advanced colorectal cancer. It's first thing he said, he thinks it's diagnosis. But the question is, we don't know why. Are there driver genes within this amplified region? Are there genes here that are required and are continuing to drive the development of colorectal cancer? So data integration focused us down on 116 genes, and then what we needed to do is I was, by this time, very sure that what we needed then was a very good model to study this in. And so they had to do QRT-PCR analysis on a very large number of colorectal cancer cell lines to find the best model that we could, where at least a significant proportion of these genes showed the same type of changes at a genetic level. They were amplified and they were overexpressed in these models. Importantly, we also need to be able to do the manipulations in these cell lines. So we need to be able to transfect them and do the siRNA studies. So from this, we got to 2 cell lines of interest that expressed high levels of 67 of these genes. We then managed to find siRNAs that were very effective to 44 of these that we felt were robust. And then what we're seeing here is a primary screen where we're looking at the cell viability for one of the cell lines, SW480s, and another one of the cell lines, DLD1. And so here's our positive control down here, where if we put this siRNA into cells, this will kill everything. But then we have others where we have 1 or 2, we just did 2 siRNAs to start with, where at least one of them induced a significant effect in one or both of the cell lines. So from this set, we got down to 17 genes where we went in with yet more siRNAs. And we got down to 8 genes of interest, which are shown here, which, to be honest, I wasn't exactly wowed by in terms of using cell viability as that endpoint. So clearly these cell lines required these genes these genes for functioning, but some of them that they were very interested in, we were only seeing very small changes. And one of the important things at this point to bear in mind that we were using a very broad phenotype. We were using cell viability, and I'm still not convinced this was the right assay, and we should have used another assay, but we still didn't know what to use. So that's one of the problems, and it's an intrinsic problem when we're looking at these things. We also saw a lot of variation between cell lines, and I now, in retrospect, realize that 2 was not enough. We should have done 4, 5, 6 cell lines. Okay? And what was critical about this study in comparison with the study I described earlier, there was no control cell line. And there is absolutely no colon or rectal cell line that is not transformed. And so we hadn't got any way of seeing whether there was a selectivity for these effects. And we were assuming single gene effects. I've just told you earlier that this is a very large amplified region with lots of genes that change, and we're only looking at one gene at a time. So we made a lot of assumptions at this point when we moved forward, but the feeling of the group at that point was that that's what we wanted to do. So we took 8 genes and decided to do expression profiling in one of the colorectal cancer cell lines to find out what it was, what was the functional space that these proteins were acting in. Because at this point, only one of these genes had previously been associated with colorectal cancer, and in some cases there was only minimal information as to the functional role of these genes. So this is what, and I'm going to blow one of these up in a minute so you get a better sense, but one of the things that my lab's done quite routinely and still continues to do is when we silence an individual gene, we then do gene expression profiling to discover in an unbiased fashion what it is that protein is doing in those cells at least at a transcriptional level. And this works particularly well when we're looking at genes that are involved in as part of a transcriptional response. And so what we're seeing here is the correlation of the gene expression profile when we've silenced a gene with one siRNA versus another. So we see very good concordance. When we've really optimized, when we know exactly which siRNAs we want to do, we get very good concordance between them. And I also really want to get you out of this, what I'd like you to get from this slide is just one thing, that sometimes in silencing this gene, we actually saw almost no changes that were significant, just a few. But this gene, we silenced this one, it's part of the proteasome pathway, actually over 1/3 of the genome changes. And we've actually silenced one gene where we actually changed half the genome as a consequence of it. So this allows us to get a sense and build hypotheses as to what a protein is doing. And I'm just going to show you here discussion of one of these, LNX2. This is a blow-up of one of the figures I just show you here, where we're looking at the fold change for one siRNA versus another. So we have some genes that don't change in the same direction, but on the whole, we see really pretty good correlation between these. And we can now use this to make a hypothesis about what this protein might be doing. So a little bit was known about LNX2's function in the normal setting. It's known to be involved in the NOTCH pathway. And in fact, if we looked at our expression profiles, we could see clear silencing. When we silenced LNX2, we saw clear downregulation of both NOTCH and NOTCH1 target genes. So we could find that, and this we could confirm at a protein level. But we were also, the whole point of this was discovery. So we were interested to see if we could find a way to look at these profiles and make new discoveries of the function of LNX2. And what Jason Pitt in my lab did and I was we went back to our expression profiles, and what we've done is we curated the targets of known transcription factors involved in colorectal cancer. For example, NF-kappa B, MYC, VDR, SMAD4, beta-catenin, and TCF7L2. So these are very key genes involved in colorectal cancer. And what we were able to show was that there was huge enrichment, particularly of TCF7L2 target genes in the signatures when we had silenced LNX2. So what we're saying here is now we have a hypothesis that links LNX2 to TCF7L2. Okay. So what is TC7L2? Well, this is required for beta-catenin and Wnt signaling, which we know is disrupted in colorectal cancer, as is NOTCH signaling. And what we were able to show is that, yes, indeed, if we silence LNX2, this downregulates TC7L2 and actually induces a change in a phenotypic reporter assay of TC7L2 activity. And indeed, we can actually go back into publicly available data looking at the expression in colorectal cancer and adjacent mucosa and show that actually the genes that LNX2 is regulating downstream of TCF7L2 show a differential expression between tumor and mucosa. So what we were able to do from this was actually link one protein that we know is amplified and overexpressed in colorectal cancer with two different pathways that we also know are altered in colorectal cancer. So I've just summarized there. In the last case study, I'm going to now move on to how we've combined RNAi with looking at some other intervention. So over the years, we've looked at chemotherapeutic agents, we've looked at radiation, we've looked at other cancer agents that act through different mechanisms. And what I'm going to talk about today is the TRAIL-induced apoptotic pathway. So what's the reason for doing this? Well, there's a number of different reasons why RNAi has proved very useful in studying drug activity, because it can give you a way of looking for synergistic molecular targets that might exploit complementary vulnerabilities. A huge amount of cost goes into looking at combination trials for drugs, because particularly now you usually have to use multiple drugs. And those are very expensive trials to do. So if we can get some sense of what would be a good combination, this may accelerate that process. It means you can use lower concentrations of the drug if there is some sort of additive or synergistic effect between them. And you can use it, and we've done a number of studies looking at it, these types of studies for looking at overcoming drug resistance. And also you have the potential of taking a drug that you know works in one context and expanding it into another cancer type. So again, this is a collaboration with Stan Lipowitz. This was work, again, started by a postdoc in his lab, Sarisha Garamella, and is now being continued actually by a graduate student in Stan's lab, Jennifer Dime. So tumor necrosis-related apoptosis-inducing ligand, or TRAIL, has been known for quite some time as a potential cancer therapeutic because its ability to kill selectively cancer cells over normal cells. And so what we're seeing here is that this is part of the apoptotic extrinsic pathway, while if TRAIL will trigger this pathway through procaspase 8 and through caspase 3-7 and trigger apoptosis. Now, the problem has been is that while you get TRAIL mediates regression of cancer xenografts without affecting normal tissues, and you actually see very good. And in fact, many triple-negative breast cancers are more sensitive to TRAIL than other breast cancer cell lines. The determinants of this relative sensitivity and resistance to TRAIL are not understood. Okay, so there have been phase 1 studies that have demonstrated that TRAIL1 is safe, but there's not been much clinical efficacy. So we need to identify predictive biomarkers that can identify which patients are going to be sensitive and which are going to be resistant to TRAIL, and if possible, identify additional genes or pathways that you can target in combination with TRAIL to enhance its efficacy. So these were the goals of this study. So what we did was we did parallel RNAi screens of the human kinome of phosphatome, that's about 350 additional genes, and we did these in the absence and presence of TRAIL, and we actually did 3 different screens. We did where we measured activated caspase 8, activated caspase 3-7, and then also cell viability. So this was a big project. I'm just going to show you here some of the controls. Again, I'm coming back to controls to be able to interpret this. What we used was 2 siRNAs to 2 proteins within the pathway, caspase 8 and FLIP. We'll go back to the pathway for a second. What we can do is this. If we inhibit this activity, caspase TRAIL caspase activation will go up, and if we take out caspase 8, if we inhibit this, we actually block this pathway, and so it will go down. So that is shown here, perhaps best here with this caspase 3-7 activation screen. So if we inhibit the effector molecule, caspase 8, we see no activation in the presence of TRAIL, which is shown here in red. But when we add FLIP, if we inhibit FLIP, which acts as an inhibitor, we can actually increase the activation. And then this is showing our real positive control that gives huge amounts of caspase activation. We see the inverse effect in cell viability. So these are rather complex slides. I'm only going to pick out one of them to walk you through, as the paper was published earlier this year. But what we were looking for here was to show that in the presence of TRAIL, well, the first thing to take from this is that when we have no TRAIL, we only see a few genes that are inducing apoptosis alone. So genes you anticipate will trigger apoptosis, such as inhibiting PLK1, are within these graphs, but we don't see much. But when we add TRAIL, we see significant activation of caspase 3, 7, or 8, and a reduction in cell viability, which you would predict. But what we were looking for here is that there is a correlation across the different combinations of the different screens. So we were just confirming internally that on the whole, the screens were acting as predicted. So we found, much to our surprise, should be said, that huge numbers of putative regulators of TRAIL. So we found 83 kinases, 4 phosphatases, and 63 genes. And this was out of a relatively small screen. And I'm just going to show you data for a couple of these things. So the open circles in black show no TRAIL. The red shows TRAIL. And what we're seeing here is that a known inhibitor of TRAIL, that when we inhibit BCL-XL, we see activation of caspase 3-7 with all 4 siRNAs very nicely. These are also known inhibitors of TRAIL-mediated apoptosis, BERK2 and BERK3, and again, we see pretty robust activation when these genes are silenced. So this proved to be a very robust screen, but we got way more hits than we anticipated. Well, we've worked through some of them for the publication, and as I say, this was published just a few months ago. This is showing some of the more detailed data for BCL-XL, BCL-XL, and FGFR4, but we see these effects pretty consistently with multiple siRNAs. What we were able to do was actually put these within a pathway, and now we've actually confirmed in multiple cell lines many of these genes as being significant in enhancing TRAIL sensitivity, including into cell lines that are actually very resistant to TRAIL. Importantly, we could also phenocopy this again with a drug. So this is where we're able to use a pan-BCL-XL, BCL2, BCL-W inhibitor, and in all cases, when we combine this with TRAIL, we were now able to actually sensitize multiple breast cancer cell lines, including TRAIL-resistant cell lines, to TRAIL. So this is now giving people a rational route for trying to exploit TRAIL-mediated apoptosis. But as I said earlier, we ended up with way too many hits, and making prioritization of follow-up difficult. And so the decision was needed, actually, to be able to prioritize it, we needed more data, because this would hopefully give us a more sense of the exact functional networks that were involved in this. And so this actually, an expanded project of this has now been recently accepted into the pipeline at the NIH RNAi screening facility. And this allows me to segue into very large-scale screening. So what I've talked about are really actually screens that are relatively small, where we're looking at maybe 1,000 or so genes at a time. Full screening campaigns are really when you go genome-wide. And to do that, as you've seen when you've visited NCATs, you have to get into big robotics, and you need to get really crazy about how you're going to do this and how you're going to analyze this. So a few years ago, a core facility was set up by my former trainee, Scott Martin, at NCATs, and I've just stolen a couple of slides from him where he's talked about their work, where they're performing collaborative genome-wide RNAi screens, predominantly siRNAs at the moment, and are trying to make as much of this available, the information that comes from these screens. These are all collaborations with investigators within the intramural program, but we're trying to move a lot of this information more broadly and the methods involved more broadly. So you can do a range of assays there. These are just a snapshot of some of the projects that are currently or have either been published or are currently ongoing in the facility. And as you can see, there's quite a number of them in the context of cancer. I'm very deeply involved in one of these in the context of Ewing sarcoma, but there are a number of others who are involved. And I keep being told I've got to point out the Ebola virus one at the moment. So these are just snapshots from 4 studies that have been published by the group, and here are included for you to go and look at more details. I've already talked about this one as the sort of a technology development, but then these are 3 recent papers from last year, where these are large genome screens that have allowed them to look at proteins involved in retinal ganglion cell death, looking at proteins that are required for poxvirus morphogenesis, and then this one that appeared in Nature earlier last year, looking at Parkin, which is involved in Parkinson's disease. As I said, we've been really trying to push that as the paper is published, that all the data is deposited in PubChem along with the sequences that are derived from that, because really the data is useless without that information. So just in the last 2 minutes, what I wanted to do was just give you some context. I've talked predominantly about RNAi because that's what my lab does, but there are alternative ways of manipulating gene expression to ask about to do functional genomic studies. So obviously, gain of function is an important other tool in our arsenal, as is other ways of inducing loss of functions such as dominant negatives, and these are methods that have been around for a while. But what I just wanted to mention in the last couple of slides is the new things that are very much coming to the forefront at the moment, which is gene editing tools that I've listed here: zinc finger nucleases, TALENs, transcription activating like effector nucleases, and cluster-regulated interspace short palindromic repeats, or CRISPR, Cas9 systems. Okay. So I've got a couple of slides here that I stole from staff scientists in my lab that actually stole them from somebody else at the University of Pennsylvania, which is just to sort of try and highlight some of the key features of these tools that are being developed. So what you're seeing here at the top one is a representation of how zinc fingers are supposed to work. So these facilitate genome editing by creating a double-stranded break. And then you can get homologous recombination or non-homologous end joining to alter this DNA sequence. So each zinc finger consists of 2 functional domains, a DNA binding domain, and a chain of 2 finger, I've got to get this right myself because I've just got to make sure, and then the nuclease domain here. So this is where you're cut, and if you've defined this sequence, you should be able to edit this gene specifically. TALENs do a similar thing. You just have a different set of domains that are used here that are based on the transcript activator sequences from a different host from Xanthomus bacteria, which is why I have it there because I can never say it properly. And then the new kid on the block that I'm sure most of you have heard of is the CRISPRs. So CRISPRs are based on, again, are an RNA-guided editing platform that uses, again, a bacterially derived protein, Cas9, shown here, and the synthetic siRNA will define the sequence where this will cut. Now, these are predominantly being used to do manipulations that you can't use RNAi for, particularly for inducing single nucleotide changes, if you want to mimic SNPs that you see in GWAS studies, for targeting long non-coding RNAs, which we cannot target by RNAi, or also just to complement RNAi studies. Libraries, particularly of CRISPRs, have now been, one has been published, but there are some issues with it. So that one of the issues with CRISPRs is that this may be subject to just as many, if not more, off-target effects as RNAi. And I've just used this slide here to summarize some of these features, that sort of compare and contrast these. I could have added RNAi onto this. the key thing about these are permanent gene editing changes, whereas RNAi will be a transient effect. And so there's definitely going to be a case for a lot of people moving over to some of these technologies to compare and contrast with RNAi. The key one at the moment is still this off-target cutting. This is still not well defined, particularly for CRISPRs. So I would urge you that if you're thinking of doing this, that you look at some of the different options that are available. And you'll see in the last slide, yeah, I've included in today's presentation the slides, some reviews that you can use to go back and sort of look at these in more detail. And these are definitely some of the best that are out there at the moment, looking at the CRISPR-Cas9 system. And it may be that this talk will start to shift over to some of these other technologies in the coming years, but I didn't think I'd make that swap today. So I've also highlighted a couple of other study papers. These are all reviews that if you're interested in some of the other features of how you can use RNAi, you can look at, in particular, this work on RNAi therapeutics, which I have included in this talk before, but it means I go too long, so I cut it today. So an enormous number of people have been involved in the studies that I've described, plus all the other studies that I have not described. I wanted to highlight now that a lot of the work that I described today involved Jason Pitt in the collaboration on colorectal cancer with Thomas Reed's lab. The breast cancer studies were all done with Stan Lipovitz, with two people in his lab, particularly Lindsay Morrow and Sarisha Garamelli, and then from my lab, including work from Sarisha Chakra, Christine Galhouse, and probably other people here that I met. And Scott Martin, who was in my lab, now runs the facility out at NCATS. So thank you.
You could go into the clinic ever so quickly.
Okay, I can try. So RNAi therapeutics at the moment are hot because there's an siRNA to Ebola that's in development. So I think siRNAs in the context of viral, inhibiting viruses is actually becoming a potentially valid approach. The cancer is hard. There is one study that came out of Caltech using RNAi against RM2 that did look very promising, but I have not seen a lot since. So that's a struggle. That's a struggle. I think where we're going to see the greatest effect moving forward, both in RNAi looking at siRNAs and in microRNAs as well, is going to actually be in the viral field to start with. We'll learn a lot from those, and then we may be able to go back to cancer and other fields. It is, yeah. So there was a paper in Cancer Discovery from a, I can't remember where the group was, that recently described their application of a WEE1. Thank you.
Okay, so in TRACO, some of you visited the tumor boards where you saw case reports. And today we're bringing the case reports to you. We have Dr. Olaku from the Division of Cancer Treatment and Diagnosis, and he's going to be doing several case report studies. So we go from the basic research back to the clinic.
Thank you. Hello, good evening, everybody. Just to talk about case reports, this is much simpler and easier than what you've just had. And what I hope to achieve at the end of this short talk is to describe the history of case reports, to recognize potential roles of case reports, to describe case reports and outline pertinent information for a good case report. This is one definition of case report. A case report is a formal summary of a unique patient and his or her illness, including the presenting signs and symptoms, diagnostic studies, treatment course, and outcome. This will give you a bit of a background of how case reports in medicine started. Probably one of the oldest examples of preserved medical literature are the ones contained in the text from ancient Egyptian papyrus around 1600 BC. And these were probably rewritten from centuries before. Among them were 48 cases discussing injuries or disorders of the head and upper torso. There is a belief that some Hippocratic physicians wrote case histories that were retrospective accounts emphasizing accurate description of only clinically relevant findings. One of the hallmarks of those case reports that focused on objective description of findings and observation of the cause of the disease. They included both mental and physical findings, although they focused more on the physical than the mental. And the patient's version of his or her complaint was, for the most part, absent. And then we come to the Galenic case reports. What appeared to be new in this case was that he added some conversational tone into the case reports. He placed himself in the context of the first person and was an active agent in the case description. For example, in his case report, he included his working day, his doubts, his tentative diagnosis, and his interactions with other physicians as the disease unfolds. Then we come to the Middle Ages. It seems at the time that medicine in the western part of the world remained dormant, and there was a lot of literature from Islamic medicine. Those case reports were similar to the Hippocratic and the Galenic case reports. And then from around the 17th to the 18th centuries, case reports adhered to the Galenic conversational tone, but they put more emphasis on patients' subjective experiences. At this time, the authors employed dramatic devices to delay the moment of diagnosis or the outcome of the story. What they wanted to achieve was to heighten the tension and degrees of physician involvement with the suffering patients. And they made the titles to be more appealing. For example, this one says, a girl 3 years old who remained a quarter of an hour underwater without drowning. And this appeared in a case report in Philosophical Transactions in 1739. And then in the 19th century, there was less emphasis on the patient's subjective accounts, and the focus was more on the clinical findings, and they started to use more technical terms. And the texts were now organized into sections, and you have, for example, the demographic details and the outline of clinical course of the events. And then they introduced more medical terminology, became more prominent. Now we look at how this also was reflected in cancer case reports. Myelogenesis described case reports as the first line of evidence where everything begins. And some of the, again, cancer case reports were traced back to the ancient Egyptian papyrus records, and that was where the first cases of incurable tumors of the breast were described. Now, what are the potential roles of case reports? One of them can be recognition and description of a new disease. For example, in 1999, in New York, you have the case of West Nile encephalitis, and the case reports and the observation of the few cases that they saw led to them being able to identify which groups of viruses were responsible for this disease and how the disease was transmitted. Another use of case reports could be detection of drug side effects, which could be either an adverse drug effect or a beneficial drug effect. Quite a number of drug retractions were due to case reports that led to investigations. And one of the success stories in the pharmaceutical industry, sildenafil, which is known as Viagra, was as a result of an unanticipated side effect of an antihypertensive drug that they were developing. And also for people who had nicotine withdrawal symptoms, the association of depression with nicotine withdrawal symptoms led to antidepressant being used to treat for nicotine withdrawal. Now, also case studies can be used to study mechanism of disease. For example, in maternally inherited diabetes associated with deafness, they looked at family histories and case report when they looked at another, went back to look at a particular family, led to the identification of mitochondrial disease, in this case, this maternally inherited diabetes associated with deafness. Case reports can be very useful in medical education and audit. In medical school, they use that a lot. It can also be used as a recognition of a rare manifestation of a disease. Recently, a colleague of mine in Minnesota published a case report of a lady that had recurrent acute pancreatitis. And one of the things he found was that it was due to an increased level of deoxycholic acid. So again, case report can be very useful. It can also be of very importance in the impact on health policy. This can be exemplified by the recent Ebola virus infection that we have here. You see how the CDC made some changes to their policy as a result of seeing what happened in the cases that came to the US. And now we'll look at how case reports are used to describe a new disease or diagnosis. Case reports started as communication between physicians, and Hippocrates and Rufus of Ephesus described melanoma cases. 1832, Thomas Hodgkin reported 6 cases, 2 of what is now what is described as Hodgkin disease. Or 1957, Burkitt described the tumor in the jaw of the African child, which is now known as the Burkitt lymphoma. And 1990, Fawcett and his colleagues described a new type of lymphoma called hepatosplenic T-cell lymphoma, which has led to more research of this new entity. So we go to examples of case reports. This is a case of a 60-year-old man who did not smoke, was diagnosed with stage 4 lung adenocarcinoma with unknown EGFR status. He was treated with ilotinib after failing to respond to first-line chemotherapy, which was pemetrexed and cisplatin. In September of 2009, patient had a partial remission in October, which was confirmed by a CT scan, and the disease progressed in January of 2010. However, his performance score still remained as 1. Sorafenib was added to his treatment with ilotinib as a salvage therapy. 2 weeks after initiation of combination therapy, the patient developed cough, dyspnea, fever, and fatigue. A chest CT scan that was done in March 2010 showed a mass in the lower lobe of the right lung with atelectasis, obstructive pneumonia, and multiple patchy ground glass opacities in the left lung. This is some of the results from his blood gas, arterial blood gas analysis. And a diagnosis of interstitial lung disease was made. The combination treatment was discontinued, and the patient had supportive care, unfortunately passed away March 2010. Now, this was the CT scan pre-ilotinib, so you can see the tumor here. After ilotinib treatment, when he had the partial remission, you can hardly see it here. Then when he had a recurrence, and this was following when they added sorafenib to it. So, This is a case of a 49-year-old lady who had stage 3 nasopharyngeal carcinoma after presenting with diffuse headaches. Patient was treated with radiation therapy for 6 weeks and concurrent bolus cisplatin, which was followed by 3 cycles of adjuvant cisplatin and 5 fluorouracil over a 4-day period. Patient was disease-free for 18 months. She subsequently developed a low back pain, and radiologic assessment identified diffuse blastic bone lesions, and a biopsy confirmed a relapsed metastatic nasopharyngeal carcinoma. She had palliative radiation to the painful sacral lesion, and they started systemic therapy, paclitaxel and cetuximab, but after that she progressed again 6 months after. Then a second line of chemotherapy was added. They added gemcitabine, and she added it on day 1, 8, and 15 for 4 weeks. Unfortunately, the disease recurred after 8 months. At that time, she had zoledronic acid with the chemotherapy, and that was subsequently changed to denosumab. And the disease progressed, and she developed axillary and iliac adenopathy. Now, a third line of chemotherapy was added. They gave her methotrexate. Again, disease reoccurred. After 3 months, she was put on a phase 1 clinical trial with a MEK inhibitor, GDC0623. Disease was controlled for 4 months. Again, she developed a new palpable painless mass in the left breast. Mammography identified as a 5.4.4 by 1.9 centimeter irregular mass at the 10 o'clock position of the left breast, a BRADS category 5. And that's the lesion there. The PET-CT scan also identified the new left breast mass, and at the time they thought it was a primary breast cancer, so which means they felt she had 2 different types of cancer. A biopsy revealed malignant cells consistent with primary breast cancer. Immunohistochemistry revealed no estrogen or progesterone receptor, and there was no amplification of HER2 expression. Tumor there, that's the PET lighting up there. So they looked at all the biopsies that they had collected previously to compare them, and they found that the biopsies were similar. The primary tumor and the breast mass were both Epstein-Barr virus positive, and then the diagnosis was changed to progressive nasopharyngeal carcinoma that was metastatic to the breast. At the time of the case report, she was doing well, and she was on a 5th line of chemotherapy. And so that just shows the similarity in the histology slide. This is a case of a 16-year-old lady that presented to the hospital with a 3-month history of a mass in the abdomen. She had acne, asceticism, and menstrual irregularities. She had a decreased appetite, shortness of breath, and constipation. On examination, she had diffuse tender palpable abdominal mass extending to the lower pelvis and abdomen up to the umbilicus. Testosterone, dihydroepiandrosterone, and CA125 levels were abnormal, and every other laboratory test was within normal limits. Ultrasound scan revealed ascites with well-vascularized mass with cystic and solid components, mostly arising from the left ovary, and a CT scan revealed a mass 13.5 by 23.3 by 21.5 centimeters. It was a complex cystic mass, multilocular and extending again from the pelvis to the mid-abdomen. There was no significant lymphadenopathies in the pelvis or abdomen. That's the mass there. She had the laparotomy, and they removed the left tube and ovary, omentum, and appendix. The ovarian mass weighed 19.1 1,945 grams and measured 24 by 21 by 7 centimeters. This cut surface showed multiple nodules separated by fibrous septum. Microscopically, it was a immature, poorly differentiated spindle-shaped Sertoli cells forming cords and ill-defined tubules. The neoplastic cells exhibited nuclear atypia and high mitotic index. So this is the specimen removed from at surgery, and this is the histology showing the spindle cells, the lytic and Sertoli cells. This one shows the mitotic figures. So they started adjuvant chemotherapy with bleomycin, etoposide, and cisplatin, and 3-month follow-up, she was fine, and there was no evidence of recurrence. This is a 31-year-old lady who had was admitted to the hospital with pain in the lower abdomen and back. An ultrasound scan revealed an 11.8 by 8.3 centimeter cauliflower-like mass noted on the left side of the pelvic cavity. CT scan revealed an ill-defined irregular soft tissue mass with extensive calcification in the pelvic cavity. 4 years prior, she was admitted in the hospital for primary infertility and amenorrhea. And on examination, she had age-appropriate normal development of the breast, PPK, external genitalia, and the karyotype was 46XS. Diagnostic laparoscopy revealed an absent upper vagina and 2 irregularly shaped uterine bodies positioned near each of the infundopelvic ligaments. Both ovaries were normal in appearance. The left kidney was small, hypoplastic, and there was a scoliotic change in the thoracic spine on chest X-ray. The only abnormal result was CA125 that was very high. The normal level goes about 35. So at surgery, they removed the 25 centimeter pelvic mass on the left side of the pelvic cavity with multiple peritoneal seeding, omental invasion, and rectal wall adhesions. Both ovaries were slightly normal, slightly enlarged but normal. So they removed everything, the omentum, part of sigmoid colon, and did some node sampling and performed peritoneal washing cytology. Both ovaries exhibited normal histological features with numerous carcinoma cell implants on their surface. The pelvic mass contained well-defined areas of ovarian tissue and cystic follicles, and immunochemistry results revealed that the serous tumor was developed from a third ovary. So unfortunately, this lady had a supernumerary ovary. She received post-operative chemotherapy 16 months after surgery. She was stable and did not have any evidence of recurrence. Pathological findings, again, the rectum was resected, showed evidence of duplicate uterine bodies with attached adnexia. The serosal surface of the uterus and both ovaries showed seedings of the tumor, the largest of which was 1.3 centimeters in diameter. Both uteri had separate cavities and blind ends, and the mass was 8 by 7 by 5 centimeters. It had invaded the uterine and rectal walls, and then the omental mass was 38 by 5.4 by 2 centimeters. So this shows us these are the duplicate uterus. So this is one uterus, this is another one. This is the ovary over here, and then this is part of the tumor, and some of the rectum are part of the things that they removed. This is the normal ovary, and all these small dots are these deposits of tumor on the normal ovary. So like I said, he had 2 normal ovaries, but the tumor developed in a third ovary that she had. So it was diagnosed as a serous papillary carcinoma characterized by thin or complex papillary growth. Both ovaries exhibited normal histological features, but had multiple deposits of serous papillary carcinoma on the surface and superficial cortex. Now we go to rare cases. In rare cases, it may be very difficult to do a study from obvious reasons that these cases are rare. We don't have too many. And case reports provide the only means by which physicians who come in contact with these patients treat the patients, because they are rare, there are no randomized trials, and all you can go by is what somebody else has reported. This is the case of a 41-year-old male who presented to rehabilitation department with a 2-month history of neck pain. He was healthy, unremarkable medical history, apart from smoking 20-pack year smoking history, which is about 20 packs a day for about 20 years. CT scan showed osteolytic changes with bony fragmentations in the C3, C5, C6 transverse processes and vertebral bodies, and that suggested that the was suggestive of pathological fractures caused by a tumor. He had a technician scan that also confirmed this osteolytic bone lesions and some fractures. Urine electrophoresis was negative. Bone marrow biopsy revealed glandular pattern of neoplastic cells that almost completely replaced the hematopoietic cells in the bone marrow. The cells were positive for AE1 and AE3. They scoped the esophagus, the duodenum, the colon, all was negative. All were negative. And PET-CT showed disseminated hypermetabolic lesion in the axial skeleton with multiple small lung nodules, and lesions in the left axillary lymph nodes. He had an ultrasound-guided biopsy of the axillary lymph node that revealed he had adenoid-cystic carcinoma. Head and neck screening did not find any area to locate the primary tumor, because typically those type of tumors are seen in this, arise from the salivary gland. Physical examination revealed a small palpable and mobile nodular lesions in the periareolar area of the left breast that was initially missed at by the CT. On this PET-CT scan, it measured about 1.7 centimeters and was BRADS category 4C. Core biopsy again revealed adenoid cystic carcinoma. Tumor was ARPR B2 negative and CK5, 6 and P53 positive. So that's C3, 5, and 6. Again, the shows osteolytic lesions and might be some chipping off of the bone there. This is not too clear, but from the TEC scan, there was evidence of fractures of the ribs. This is some of the lymph nodes here. The lungs had nodules, and that's the breast mass there. This is the histology. This is from biopsy, bone marrow aspiration, sorry. C5 to F is the bone marrow biopsy, and this is evidence of the tumor in the lymph node, evidence of the tumor in the breast lesion. This is another case of a 23-year-old male, is the primary osteosarcoma of the heart. Presented with the clinic with a history of chest pain, cough, and hemoptysis. Chest X-ray revealed a left pleural effusion. The cytology from the fluid did not reveal any evidence of malignant cells. However, he had an echocardiography that revealed a left atrial mass measuring 3.8 by 3.8 centimeters in diameter and filling the whole of the left atrium, and extending into the mitral valve during diastole. So that's the mass there in the left atrium. The presumptive diagnosis was a cardiac myxoma. The tumor was excised and he had a valve replacement. Pathologic examination revealed a high-grade conventional osteosarcoma arising from the cardiac wall, and it was mostly osteoblastic in nature. bone scan and CT scans were normal. And he had adjuvant chemotherapy with cisplatin and ifosfamide was given for 6 cycles. So after the chemo, the thoracoabdominal CT and echo were normal. 9 months after the last cycle of chemotherapy, he presented with left hemiparesis and abnormal speech. CT scan of the cranium showed a central pontine mass 3 by 3 by 3 by 2.5. And that's the pontine mass there, hypervascular mass with some calcification there. The radiologist diagnosed as a metastatic lesion. Surgery could not be performed because it was close to very vital nervous structures. He was given radiotherapy, and he was treated with radiotherapy. And during the course of his hospitalization, he lost consciousness and he died because the tumor had compressed vital structures on the brain stem. And that was what was removed from the left atrium. So this is an irregular mass with discrete margins. And this is, again, histology from the osteoblasts. It's osteoid. This is a 64-year-old male patient that was admitted with a history of progressive dyspnea and cough. He had initially undergone a lobectomy of the liver for hepatocellular carcinoma in 2006. 14 months after, CT scan revealed multiple metastases and left adrenal gland metastasis. Serum protein induced by vitamin K absence or antagonist 2 level was elevated. Normal is up to about 40. And he was given palliative chemotherapy with 5-fluorouracil, adriamycin, and carboplatin. Progressed following 2 cycles of chemotherapy. He received 2 more second-line chemotherapy with 5-FU, etoposide, and carboplatin, and 3 cycles of third-line chemotherapy with oral capecitabine. There was no objective response. Chemotherapy was discontinued, and patient was transferred to another facility in 2009. Of note is that he had a history of hepatitis B virus infection in the past. And some of his lab results just looked normal. Alpha-fetoprotein was slightly high, and chest radiograph revealed miliary nodules in the lungs. CT scan of 2009, February, indicated miliary metastasis and metastatic abdominal lymph nodes. Now, this is the chest X-ray. You can see all the lungs with nodules. CT scan. He had at this new facility, he had follow-up every 3 months. And he continued to have cough and dyspnea, and there were no changes on the chest radiograph. However, the second visit in September 2009, his general condition had improved, and the cough and dyspnea had stopped. And the chest radiograph revealed that all metastatic nodules had disappeared. So this was in September. All those nodules had disappeared. The serum alpha-fetoprotein had decreased to 1.5. Whole body PET scan in December of 2009 revealed that all metastatic nodules in the lung, as well as the lesions of the adrenal glands and lymph nodes, had disappeared. However, the patient took herbal medicine, dendropanax, mobiferralivir, for approximately a week, apparently recommended by his family. And follow-up chest and CT scans did not reveal any recurrent metastatic lesions. And all the tumor markers were normal. And as of May 2013, the patient was alive and well without any tumor. So chest X-ray, February 2009, showing the nodules. June, the nodules. September, disappeared. 2011, 2012, 2013, all clear. supposed to be spontaneous regression, but it's difficult to, difficult to say. But because it took whatever it took for 1 week, can we say that played a part? We don't know. This is a 58-year-old lady who was admitted to the orthopedics department July 2012, 6-month history of left plantar heel pain. This was aggravated on walking and standing, and she had been diagnosed with chronic plantar heel pain 6 months earlier in a local hospital. The pain wasn't relieved by any anti-inflammatory or analgesic treatment. She didn't have any fever, no radiating pain to the lower leg, no night pain or numbness to the local skin. Examination revealed alteration of the skin color and soft tissue swelling around the left calcaneus, and there was tenderness at the end of the plantar fascia below the heel. There was no paresthesia of the surrounding skin, and the range of motion of the foot and ankle were normal. Patient was healthy with no past history of smoking, coughing, or hemoptysis, and the foot and ankle radiograph showed spurs in the left heel and an osteolytic lesion in the left calcaneus. CT scan revealed an space-occupying lesion of the calcaneus with bone destruction, and the chest CT revealed a lesion in the posterior segment of the right upper lobe. Biopsy showed a poorly differentiated adenocarcinoma with all those things positive, and patient had the left calcaneus lesion curettage and replacement with bone cement. She received palliative care but succumbed to non-small cell lung cancer 6 months after diagnosis. Again, you can see osteolytic of the bone there and the lesion there. This is the CT scan. That's the lesion. There's osteolytic bone of the calcaneal bone. This is an MRI, again showing the lesion there. And this is a CT scan showing the chest, the lung cancer. So she presented with a heel pain. And it was from investigation and the biopsy from the lesion of the foot that led them to find that she had lung cancer, even though she didn't smoke anything. And unfortunately, she succumbed 6 months after that. So this is the histology, tumor cells with large nucleus and all these fibrin strands. This is tumor in the blood vessel that shows that there was metastasis. And these are just some tumor markers staining. So our case reports relevant in this day of evidence-based medicine. Some people feel that case reports shouldn't be reported in journals because it doesn't contribute so much into evidence-based medicine. And it's obvious that from a single case report, you can't determine the safety and efficacy of a particular intervention. Albrecht and his colleagues published 100 case reports in the archives of dermatology, and they had concerns, and one of it was that there was publication bias towards positive results, exaggerated claims of efficacy and safety, inadequate informed consent reporting, under-reporting of patient-centered outcomes. The challenge is from a business point of perspective, case reports are rarely cited, and that creates a dilemma for journal editors because it can negatively affect the impact factor of the journals. The other issue is that when people are planning to publish, they will probably want to publish in a journal with a high impact factor, which in turn could influence where companies determine which journal they want to advertise their drugs or devices in. So evidence-based medicine essentially trying to find the best clinical evidence, whether we should apply a particular therapy or test, a diagnostic test to a patient. And what randomized clinical trial does very well is to evaluate therapies or tests, especially when we don't know which one has a better or the best clinical value. So case reports have their place, and it's also important in the progress of medical science and education. Mayfield and Gorin looked at 21 cases of tamoxifen-related ocular toxicity in breast cancer patients. As a result of this review, it led to having a clearer picture on the nature and distribution of this toxicity, as well as the severity of the ocular findings. It also highlighted the difficulty in trying to attribute the ocular toxicity to either tamoxifen or other things that can cause ocular problems like retinal, macular, and corneal abnormalities. Nordin conducted a 20-year literary review, again looking at primary carcinoma of the fallopian tube. As a result of that, he was able to characterize, have a more appropriate patient characteristics, be able to, and also be able to hypothesize on the underdiagnosing of the cases, what are the causes of treatment failure, and the absence at the time of lack of control trials and usefulness of a second look laparotomy for monitoring disease response to treatment given. We looked at case reports in cancer patients who use herbal therapy. 18 said was a positive outcome, 21 were on toxic effect. At the time, there were clinical trials of some of these herbs like green tea, phytoestrogens, mistletoe, ganoderma, lucidum, noni, silymarin, and others. But what we found that many of the herbs with promising case report findings have either not been explored, or maybe they are not written or reported in journals that are written in English. So we know that from this review or a systematic review of case reports, that what we can get is the characteristics of the cases and the case outcomes. However, in situations where it's impossible to conduct clinical trials for whatever reason, either because the numbers are small or for ethical reasons, the best of the first line of evidence should be taken into account. What impact has case reports have on medical research? When we look at this, case reports that were followed by published trials, only 17%. This was looking at 64 case reports. And out of that, when you look at the outcome, 27 said it was success, 60% was improvement, and then 5% was failure. And when you look at the case series, again, case reports that were published, followed by published trials, 31%. This is more now. That's not surprising because it's on case series. But when you look at the reported improvement or cure, 79%, treatment failure is 10%. So if case reports are going to be useful in clinical research, how do we write a good case report? Because if we can write a good case report, then it may provide a template in which to conduct clinical studies. Last year, the CARE guidelines were published, and CARE means case report guidelines, by a group of people who was trying to find a way to bring some sanity into case report publication. And these are some of the things they suggested, that in writing a case report, it should have a title, keywords, there should be an abstract, introduction, patient information, clinical findings, have a timeline, like either have a figure or a chart that outlines what happens when the patient was seen, say for example, the year or the month and what investigations there are. So at a snapshot, one can take a look at it and have an idea what happened. Have diagnostic assessment, what therapeutic intervention the patient had, what was the follow-up and outcomes, have a discussion. But they also believe that it should include the patient's perspective. There's some controversy about that, and informed consent. So we'll see with time if journals will begin to require this list of things from the publishers of case reports in the future. So time will tell. So case reports should be written in an organized and structured manner. It is still relevant in today's medicine. In fact, over the years, there's been a lot of, there have been some journals that are specific for case reporting now. So you have case reports in medicine, you have oncology case reports, surgical case reports. And just in the last few weeks, the Journal of Canadian Family Practice decided to start publishing case reports again. So it seems to be, again, there's a swing back to some recognition of case reports. But in rare instances where clinical trials are not possible, either because the numbers are not there or for ethical reasons, then case reports may be the only evidence available to recommend treatment to patients. Thank you. I'm just prouding my colleagues, Dr. White and Dr. Farazia. Thank you. Any questions? I told you it was going to be easy. Yes, ma'am. Currently, it will be the clinicians. We are hoping that case reports will become a template where it can generate some hypotheses to now conduct clinical studies, especially in this day and age where there are more targeted and personalized treatment. So it may become, and that's why having a standard would help, like consult and the other things in the other areas, so that you can look at 200 cases and you're able to get a good information out, rather than what we have now, people just writing the way they feel, and each journal has their own standards. Any other questions? Thank you very much.